So next up, I think we have Professor Viive Howell just giving us an update about the Biobank. Viive is the head of the Walsh Lab.
Hello everyone, my name is Viive Howell and I'm the academic lead for the Colling Institute Tumourbank. So what is tumour banking? At its most simplest, it is the collection of specimens and the later distribution of these for research. As you can see here though, there's a lot of other steps involved. Why is it important? Well, we've heard a lot about discovery research today, and it's essential. It provides the specimens for discovery research. It provides the specimens for the validation of discovery research. It links basic scientific and clinical research. It provides the specimens to develop new models for therapeutic testing in vivo. And all of this contributes to translational research and research training. So what does it involve? I've shown you some of the steps. So clinical engagement and surgical engagement is absolutely essential to keep the pipeline going so that we can collect these specimens. It's imperative that we have trained and dedicated staff. We need infrastructure such as centrifuges, freezers, fridges, computers, databases. Appropriate and up-to-date ethics and governance is absolutely essential, as are standard operating procedures and certification from New South Wales Health. And our Tumourbank was only the 5th in New South Wales to get full certification from New South Wales Health. Just one recent initiative that used all of these procedures was the establishment of remote consenting and paperless consenting in this year of the pandemic. Additionally, data security is absolutely paramount to protect clinical information, and all of this needs to be behind a New South Wales Health firewall. And finally, of course, none of this would be possible without funding. And here I'd like to acknowledge funding from Sydney Vital for several salaries that has been going on since Sydney Vital was established. So what has the Tumourbank done? It was established in 1992 on this campus. And in terms of activity, it first started by collecting rare endocrine tumours for research. This has since grown and it now collects tissue and fresh tissue and blood from approximately 6 different tumour streams. Over this time, it has accumulated over 35,000 specimens from 11,000 patients. In terms of how to measure success, we could do that with PhD completions and publications. And over this time, the Tumourbank has contributed specimens to the successful completion of over 50 PhDs. These students are now professors and clinical researchers on this and other campuses. And these projects have resulted in well over 100 publications. And all of these are aimed at improving outcomes for cancer patients. Now, this is an underestimate of publications because we rely completely on the researchers to let us know, and they often don't. I'd also like to point out that with the changing time frame for PhD completions at the University of Sydney being restricted to 3 years, such research, discovery research into rare and uncommon cancers will simply not be possible if we don't have tumour banking. Just to give you one example, my most recent PhD student had to look through 15 years of the collection to find just 6 matched pairs of a rare brain cancer that ended up being the foundation for her research. Without this facility, without this capability, that research will simply not be possible. So what is the impact of this? So my own PhD student resulted in changes to the WHO guidelines for the diagnosis of parathyroid cancer. All of this discovery omics research leads to increased knowledge and understanding of the molecular basis of cancer. This has improved subclassification of a number of cancers, such as medulloblastomas. It has also allowed better prognostication for cancers such as endocrine and brain cancers, leading to increased quality of life, as well as in pancreatic cancer, leading to increased survival. And through all of this increased knowledge of the molecular basis and the driver mutations for tumour development and progression, new therapies are being developed. So in summary, tumour banking is integral and essential to research, translational research on this and all academic hospital campuses, not only locally, but around the world. The Kolling Institute Tumour Bank supports research training. It supports the establishment of clinical centres of excellence. And this activity has resulted in improvements in medical guidelines, improvements in prognosis and diagnostics for cancer patients. Thank you.
Thanks very much for that, Viive. Great presentation. Any questions? Alexander. You're on into talk time, not?
If you had to make one wish and it would come true regarding the tumor banks, how will you make them more effective?
Well, you'd give them funding for starters. It's all around money, isn't it? Everything is around money. Look, they're being used a lot more. They need to be integrated better. So, for example, we need to have them become more sustainable. And we can do that by linking them with clinical trials, because the staff that we have are certified to do all of the consenting. They know what they're doing. We should be incorporating those into prospective studies around the campus. We should be incorporating those into clinical trials rather than reinventing the wheel and having people being taught how to do this and then using that for some of the sustainability. We can be looking at how better to combine with other banks that are around. I mean, every other person is making their own bank. We don't know why. We need to look at why they are doing that and try and bring them all together into one, so that we don't have 5 people doing the same thing for 5 different tumour streams. There's some of the things.
Thank you. Another question, Anna?
Thank you very much, Viive. I would like to ask, just from the practical side, how the access to the samples can be organized for the external researchers. This is one question. And the second question is, do you have digital copies of the samples? For example, do you have scanned histology or some, I don't know, omics data or whatever? Thank you.
So sorry, I couldn't quite get that. So the first one was about access. Right. So you, to access specimens in our bank, you need to have a valid human ethics protocol that includes it, and then you apply to our tumour bank. It goes through a process which is really quite easy, and then you can get specimens. You really need to know what we've got first to make sure that it's going to work for you. In terms of what we have, we don't have that much, and often it's up to the researcher themselves to find that information, and that is a flaw. We have some funding from Sydney Vital to start to do some of that quality control, and we've been doing that for brain cancer. And so as part of that, what we're doing is having a H&E made, and they're being looked at, they're being assessed, and especially if you know GBM, there's a lot of necrosis, so we know that some of those specimens are going to be no good. We can toss those out. All of those slides will then be digitised, and they will then be available so that the researchers can see those. But that's only a relatively new initiative, and it's, without a lot of funding, it's slow going. But we have done that for, I think, maybe about 200 of the GBMs that we currently have in the bank. But then we can also make arrays from those. So we don't have it for a lot, and it really depends on the tissue that you're after as to what sort of pathology has been done on it. We do have the diagnostic report as well accompanying every specimen. But for clinical information, for omics, that's very much up to the researcher.